is to deal with what's called the central dogma. Which is when Francis Crick of Watson and Crick proposed this, there was not a whole lot of evidence for it. So it was a thing that you kind of took on faith. It would have been in the late 50s, early 60s. And then over the course of the 1960s, this got worked out and turned out to be actually true. So it's now no longer something you have to accept dogmatically, but in fact, there's tons of evidence for it. So basically, the question was, how does DNA, or I'm going to put it this way, a gene which is made of DNA, make a protein? So Watson and Crick and those folks who worked with them knew genes were important. They made proteins. Previous evidence had showed that. It was not at all clear how this worked. And the answer will turn out to be base pairing, right? As we'll see. That's how this process works. And it's a rather step-by-step thing that we'll take you through. So I want to give you an overview, and then we'll get into details. So you've got a gene. You start with a gene, and you end up with a protein. And this is information flow. That is copying and translation, but not chemical conversion. So I want to be really clear about this. The problem with this course, one of the many problems with this course, is I use arrows for all sorts of things. This arrow up there means information flows. So when I say gene makes protein, it's not that I'm taking a gene and making it into, I'm converting it into a protein. Like sometimes those arrows, like glucose is converted into pyruvate. I'm not converting a gene into a protein. I'm using the information in a gene to assemble a protein. It's like you got a bunch of directions for making some gadget, Lego project or like, and then they use that information to construct the project. So the gene is information that is copied and translated to make the protein. And so there are a couple of steps. So genes are made of DNA, and this is permanent in cells. And DNA is first copied into what's called mRNA, that is messenger RNA. And this is temporary. Talk a little bit more about that later. And then that mRNA is used to make protein, which is also temporary. All proteins last for a certain amount of time in cells, and they're degraded and reused. And the protein here does the biochemical functions. Things like, eye color, PKU, etc. All right. So there's a couple of steps I want to talk about here.